Hi, my name is Darren, along with my colleagues Andrew and Cherie, and today we're going to be discussing how Parkinson's disease impacts brain morphology. Today we're going to be discussing a literature review that we conducted to better understand the mechanism of Parkinson's disease, or PD, and then we'll be ending with questions going forward as well as concluding statements of our research.
Parkinson's disease is the second most common neurodegenerative disease following Alzheimer's, and since it can be developed over time and is the progressive loss of function or structure in neurons, we want to determine if there are any predictors for this disease, how it can be treated, and how it impacts the brain structure.
Within our literature review, we discovered something called tauopathy, which is a large component to the progression of Parkinson's disease. There's something called microtubules within the structures of the brain that are small polymer structures that help maintain the structure of the cell and regulate the synapses within the brain. And there is the presence of tau proteins that integrate with these microtubules to provide strength and stabilization to maintain the physiological conditions of our microtubules. For a not yet well understood mechanism, the tau protein becomes hyperphosphorylated or misfolded. This abnormal tau protein now starts the process of disintegration and it causes neuronal dysfunction.
So hypophosphorylated tau proteins will lead to the formation of neurofibrillary tangles, or NFTs for short. With the formation of NFTs and disintegration of the microtubules, this will impair the transport capacity of axon tubules and eventually contribute to cell death.
In order to promote aggregation and the formation of fibrils, these misfolded tau proteins will interact with the alpha-synuclein, which will then lead to abnormalities associated with axonal transport as well as the development of Lewy bodies.
Further research has shown that Parkinson's disease primarily starts in the substantia nigra, which is the area responsible for regulating the motor functions. And it then progresses through the networks within the brain as the misfolded proteins can transmit their shape through cellular transfer.
All right, so for questions going forward and possible directions for upcoming research, we can look at the hypophosphorylation of the tau protein as a good way to detect Parkinson's disease early. So research can look at how we can improve the early, diagnostics for early detection. We also saw in other research that the genes can play a role in PD. So to what extent are genes at play? And finally, what new therapy treatments are coming available?
So in conclusion, Parkinson's disease is a prion-like disease that results in brain atrophy, and baseline and 1-year MRI scans are very important predictors of disease progression.
This concludes our presentation. Thank you for listening.